Welcome to Discover ASAP, the video series where we highlight the top findings and tools connected to the ASAP network. Today, I am talking with Zack Gaertner, an MD PhD student from Team Awatramani. For today's episode, we're going to be discussing the spatial transcriptomic dataset from Zack's paper, Molecular and Spatial Transcriptomic Classification of Midbrain Dopamine Neurons and their alterations in a LRRK2G2019S model of Parkinson's disease. So Zack, to get us started, can you tell us briefly about your team's ASAP project and how this data set relates to that project?
Yeah, absolutely. And thank you for having me. So essentially, Team Awatramani's long-term goal is to look at the molecularly defined subtypes of dopamine neurons. These are dopamine neurons being categorized by unique genetic signatures that can separate them from other dopaminergic neurons so that we can isolate them into different groups. And the long-term goal of the project is to look at the different subtypes, both in terms of their inputs, outputs, their roles in different functions, as well as their roles in Parkinson's disease. So for instance, which ones might be selectively degenerating versus spared.
Great. So I think this is a really important and interesting question for people who are interested in sort of cell heterogeneity in general and in Parkinson's in particular, given the sort of specific vulnerability of different dopamine neuron subpopulations. And I know this is something that you delve into in great detail in your paper, but I want to kind of start high level and ask you to give us a brief overview of your data set and sort of the main clusters that you were able to identify.
Yeah, so essentially what we did was create a large data set of single nucleus RNA sequencing profiles of dopamine neurons isolated using a Cree line that expresses a nuclear bound GFP only within dopaminergic neurons, after which we sort them using a microfluidic sorting protocol to isolate just those cells and build single nucleus transcriptomic profiles of them. Afterwards, we then took this RNA sequencing data and combined it with another data set that a collaborator had been working on, Jean-Francois Poulin at McGill, using MERScope, so doing in situ transcriptomic profiling, so that we can figure out where do these clusters actually map out within the mouse brain, what's their distribution, and with that, how can we sort of tie this together with prior literature that's seen things like selective degeneration of ventral dopamine neurons compared to more dorsal ones. So as far as establishing the subtypes and what do we find, what we've really been able to do here is take what has been described in the past, a lot of the so-called classic subtypes of dopamine neurons, and break them apart into more granular resolution, finding new subdivisions within them. So in our paper, we separated out dopamine neuron subtypes into three broad families. One defined by the transcription factor SOC6, another defined by expression of CALB1, and a third one with expression of GAD2. Within these families, which have all sort of been previously described in various papers that have looked at profiling of dopamine neurons, we found a lot of new individual subtypes that we were able to then map to very distinct anatomical distributions and, in some cases, vulnerability in Parkinson's and some more ongoing research in our lab.
Great. Yeah, I think that one of the things that's most exciting about this paper is that you were able to identify heterogeneity beyond, as you say, those classic anatomical clusters. And so I'm wondering if we can take a minute here, even though this is really about the data set. I think the technique that you used is also important. And so I want to ask you to expand on how you were able to observe beyond, clusters of cells beyond these classic subpopulations in this paper.
Yeah, so this was sort of born out of many, many months of troubleshooting other protocols trying to do single nucleus RNA sequencing of dopamine neurons. So we had previously published a data set in Nature Neuroscience last year where we had used FACS sorting to isolate dopaminergic nuclei very similarly to in this paper. The big issue with those types of methods is, number one, you're putting a lot of stress on these nuclei. They're being ejected through a FACS machine at extreme speeds and pressures. You end up rupturing a lot of the nuclei in the process. And it takes a lot of time. And unfortunately, as that time is passing, you are losing data quality. So you are getting further and further away from sort of the ground truth of what RNA was present in that cell at the moment that we sacrificed the animals. So what we did was move to a new method that uses a microfluidic chip using the MaxQuant Tito sorter, where essentially it's able to sort these cells in a very similar way to classic FACS-based sorting, but by channeling it through a very, very small, narrow channel where the nuclei pass single file, and there is a physical lever that pushes them to either the output we want or the discarded pile, so to speak. The advantages here are it was much, much faster than what's been done in the past by sorting these using classic FACS methods. And it's much more gentle on the cells. So the actual pressure that is placed on the cells is dramatically lower. And so we found just massive increases in our yield compared to our old protocols when we did it this way. So with that, we were able to increase the size of our data sets by almost a full fold change. And not only did we increase the number of neurons that we were able to recover, but the actual quality. So the number of UMIs, unique molecular identifiers per cell. So essentially the number of RNA molecules that we were able to capture in each one of these cells was dramatically higher than what we had previously done and what a lot of other papers have been able to accomplish. So with that, we were able to get really, really nice resolution among our subtypes. And the other part of it that I think was sort of unique to our paper that really helped as well with the separation of these different subtypes was combining it with the MERScope data. As some of these populations are pretty difficult to distinguish on the basis of gene expression alone. They have very similar transcriptomic profiles, but their localization within the mouse brain can be very distinct. And so as sort of a long-term vision for these methods, we see this as being one step towards iterating more and more information onto what do we know about individual cells so that we can separate them better. So for instance, within cells that to us at the moment look like a single cluster, is it possible that there's very distinct projection patterns within those or inputs to them that we could then separate these into different functional circuits even on the basis of information that hasn't yet been captured?
Great. Yeah, I agree. I think this is a really powerful technique with a lot of future, really interesting experiments to come in the future. So you have alluded to some of this a little bit in the conversation thus far, but I would be remiss if we didn't talk about the Parkinson's aspect of this data set. And so I want to ask sort of a very broad question here about what you feel that this data set contributes to the Parkinson's field. So you can talk about, what are the specific clusters that you found impacted in Parkinson's, and how do you think that what is what are the next steps for you as far as chasing down the role of these subtypes in PD?
Absolutely. So I think there's two main points that we've really been able to make with this paper that I think are very relevant for PD research moving forward. One of which is looking at what actually is changing to the transcriptomic profiles in these cells. So what's changing at the level of gene expression in these cells in the presence of a very common Parkinson's associated mutation, LRRK2G2019S. So what we did for this was create data sets in mice that were both of wild type strains as well as those that had a knock-in copy of the LRRK2G2019S mutation, so that we can cluster by cluster compare what is actually changing, which clusters are changing, is it all of them or just some of them, and what's sort of the downstream effects of this mutation that we know is risky for causing PD. What we found was pretty interesting, and it would take quite a while to try and summarize everything in the paper, but essentially we found a lot of enrichment of pathways related to different metabolic functions and various other things that have been long associated with mechanisms of PD degeneration. On top of that, we also were able to look at cluster by cluster using our sort of high resolution data set. What could we say about which of these clusters might be vital for Parkinson's disease? One way that we approached this was looking at enrichment of GWAS-associated Parkinson's genes in each cluster. And again, what we found was similar to what's been described in prior studies. Subtypes that localize to the ventral tier of the substantia nigra seem to have a very strong enrichment for a lot of the genes that have been associated with Parkinson's disease pathogenesis. On top of that, one cluster in particular that has now become a very big interest for our lab is dopamine neurons defined by the gene ANXA1, annexin A1. So this is a subtype that in our previous paper in Nat Neuro last year, we showed represents the ventralmost subset of substantia nigra dopamine neurons in mice. And what we found was that this particular population has by far the largest enrichment of Parkinson's associated genes. So this falls in line nicely with literature showing, again, ventral specific degeneration in Parkinson's patients. With this information, we think it sort of unlocks a lot of potential for what we see as the long-term goal of this type of research, which is if we can understand the differences between these subtypes and we can understand why some degenerate but not others, that gives us a lot of information as to how can we prevent that? How can we intervene? What is the specific things that's making it vulnerable? And how can we leverage that information to prevent degeneration in Parkinson's disease?
Yeah, sure. And I think one of the things you talk about in the paper is you make a distinction between cell type and cell state, and sort of how you think this might relate to the understanding of the pathogenesis of Parkinson's disease. And so can you talk a little bit about that here and explain, what you mean by that, what you saw in the data, and and how you think what the significance of the distinction is.
Yeah, absolutely. So this is sort of the grand criticism to this entire view of these molecular subtypes of dopamine neurons is, how do we know that these are really truly distinct cells versus just different states that any given dopaminergic neuron could be in at any given time? So for instance, when we see a pattern of different genes being expressed, is there something inherent to that cell where that pattern is rigid, that pattern is encoded in this cell such that it is unchanging across its lifetime? Or is it merely responding to something that is happening in its environment, whether it be it had been particularly active that day, or it if you had encountered a toxin or any other thing that could alter the gene expression pattern in these cells. So we wanted to show in this data set that if we're breaking apart dopaminergic neurons, that all of which are extremely closely related, and the vast majority of their gene expression is the same. If we want to break apart these very, very closely related neurons into 20 or potentially more subtypes, are these real, essentially? Are these really subtypes? Or are we looking at slightly different patterns of gene expression that you'd expect to just happen transiently? What I can say is from our data, it appears that these clusters are rigid, so to speak. So we see across all of our different samples, the clusters emerging in equal proportions and equal, in roughly equal amounts. And specifically in our mice that we had given the LRRK2G2019S mutation, we show that even in the presence of this mutation where lots of gene expression is altered, the subtypes themselves are not. The actual subtypes come out exactly the same. The proportions come out the same. When you take this together with other experiments that have been done in the past looking at cell fate mapping of different subtypes of dopamine neurons, it, I think, becomes very convincing that these subtypes are likely very rigid and encoded even at the level of the floor plate progenitors.
Yeah, I think that's fascinating and a really important distinction and I think really interesting to see how in your data, how the Parkinson's mutation sort of lays on top of these identified clusters. I think that's really fascinating. So I am going to give you the opportunity now to talk about anything else that we haven't talked about that you think is important before we wrap up.
Sure. And I think you'll be talking to another one of my colleagues about the tool that we built for this paper, DopaBase, which I guess I'd like to just preface sort of our vision for this, where it's going in the years to come. So essentially, we are making a portal that allows any lab to explore our data set, both in terms of the RNA-seq information, but also now looking at the MER-scope, the physical distribution of these different cells across the mouse brain. And we see this as really just the first step of integrating as much information as we possibly can into one cohesive data set about defining these different subtypes of dopamine neurons. So for instance, can we integrate into this same information, the same data set, the projection patterns of each individual subtype, or using rabies tracing, the inputs to each individual subtype, or using things like GCAMP, the functional activity profiles of each individual subtype, such that we can sort of amass this one universal data set for people to explore, where you have all the information there is to be gathered about these different subtypes of dopamine neurons in all aspects of the properties that make them unique. This is, of course, a work in progress that will be over years to come, but the earliest version of this, with at least our RNA-seq data as well as the spatial transcriptomic data, is coming out with this paper.